Okay, welcome everyone to my talk on the poster about CRISPR knockin designer. And this is software for automatic oligonucleotide design to introduce point mutation by CRISPR-Cas9 and its application. So this is for ZDM15 conference taken part in Sheffield, but I'm attending it online. So the basic strategy of knockins consists of designing an oligonucleotide, also called single-stranded oligodeoxynucleotide, with particular mutations. So usually the desired mutation and some extra mutations to inactivate cutting by Cas9, such as PAM or site mutations. And then say you have a target exon, and then you have your Cas9 and guide RNA, and then you have the cleavage by Cas9, and then homology-dependent repair by this oligonucleotide. Unfortunately, in many model systems such as Zebrafish, this technique only occasionally works like really well, so there still needs to be some improvements. And the one side of the improvements that can be made is in the design process. So most typically people do the design manually, and there are some limitations to this. So this is quite tedious, takes a lot of time, and since there are many manipulations, and you need to sometimes you need to switch between different CRISPR types, which is hard, and then it's almost impossible to iterate over many options that sometimes you want to find the best additional mutations. And then this requires usage of separate software programs and additional manual or special computational screening of the results. And this is difficult to scale, obviously. However, there are advantages of the manual approach. Then you develop a high level of understanding of the mutation you're trying to introduce, and then you almost automatically carry out the additional verification of the introduced mutation. So the software I'm going to talk about is called CRISPR knockin designer. So I wrote it in the R Shiny environment, and this is helps toward the increased automation of oligo design. And in some ways, this is extension of the manual approach, based at least on how I do it, and I try to handle different situations automatically. And then this still requires initial test of guide RNAs and some primers. And then so what I developed is web-based implementation of using Shiny R package and other bioconductor tools. And this looks approximately like this. So this is the input elements. So this what mutation you want to introduce, then what kind of sequence you want to introduce if you do the sequence-based story, or okay, so, or you have, or you, and then you also define some guide RNA parameters. Then this is the algorithm overview of the program. So then you have the gene mutation, and then the guide RNA input data, primers, and then some oligo options, how you want your oligos to be made, so such as homology, arm length, orientation. Do you want to mutate PAM or guide RNA site? Do you want additional mutations, silent mutations for restriction enzyme sites? And then you have options for like automatic data entry based on ensemble transcript IDs, or you can do completely manual data entry of all the sequences which are relevant. And then you apply the CRISPR type option, so which CRISPR type you want to use, and this is like the main two types which are in routine experimental use. They're being used in the program, and then you select and run design pipeline. So just a note on the genotyping. So in this software, and like in general for knockin, for the knockin process, I consider two approaches: allele-specific PCR, so where you design a primer matching either the mutant or wild type allele, and the other primer is so-called site assay, forward or reverse primer, and they would be common to both of these assays. However, if your difference is sufficient, only say, and you have the wild type sample, your knockin specific primer shouldn't work, and if you say isolate a pure mutant with the knockin allele, then your wild type primer shouldn't work. I mean, there could be some exceptions and some non-specific amplifications occasionally. So this to deal with this kind of problem, you can do an alternative way. It's called restriction-based genotyping. So when you have the two site assay primers surrounding the mutation site, and then you do the digestion, and if the mutation is present at sufficient frequency, then you would get a nice result. Okay, then in terms of design of additional silent mutations to inactivate PAM or guide RNA and introduce restriction site, we usually do it the following way. Say, then if you have your target codon, then we find which codons like overlap PAM or guide RNA, and then you find potentially candidate codons inside the guide RNA site, and then we search for mutations which can help inactivate them. And then for restriction site insertion, we only consider the 3 codons nearby to the target codon, and then try to mutate each codon synonymously and check if this mutation introduces a restriction site. Okay, then the output looks approximately like this. So the first part is visualization of the targeting strategy, and then these are like the result buttons to download full reports, and then these are an overview of the results for the basic quality design. Then another thing that we considered in this program was the mutability of codons straddling 2 exons, and there are like certain intron classes called 1, 2, and 0. Phase 1, phase 2, phase 0 introns, which either split or do not split the codons, and then you have the corresponding exon classes. So if you have a situation like the phase 1 +1 to, and like -2 here, then and you try to mutate from a particular codon to a particular like amino acid, this fairly sparse matrix indicates that it's far from easy and not always possible. And the better situation is when you have like phase 2 intron, where you have +2 and -1 here, and then you much more easily can mutate, but also not always. So the program kind of takes care of those details, so you don't necessarily need to consider them explicitly. Yeah, and these are a couple of examples of experimental applications. So because these projects are ongoing, I'm not disclosing the exact mutations, but the basic idea that they were like oligos designed with the program, and then we injected them with Cas9 protein, and then performed first allele-specific PCR here. And then for this particular case, we also performed the enzyme digestion. So enzyme digestion is much less sensitive, so we only got some results in like 4 embryos. And then for this knockin, we so far only performed the knockin specific assay. Okay, and then finally, what could be like the applications of this like CRISPR mediated knockins? So now people kind of to increase efficiency, considering alternative strategies, how say the oligonucleotide or single-stranded DNA can be attached to the Cas9 or Cas9 RNP complex, and you can do it by cRNA. It says DNA chimeric oligos, or you can attach to tracker RNA. I mean, this one is much more expensive. This one is a little bit less expensive, and there is also emerging some additional strategies, such as when guide RNA has a special aptamer region called S1M, and then streptavidin binds to it, and then you can attach a biotin to your oligo, and this would also provide an opportunity to bind your oligo to the Cas9 RNA. And then the PEG RNA is, so for the prime editing, is like an emerging technology. It's not being used in zebrafish that much, but we incorporated some tools to design the outputs, which can be used as an input for PEG finder. And this may help to design prime editing strategy using our tool and the specialized prime editing tools. Okay, thank you very much for your attention, and I would like to thank all my collaborators and lab members, and I'm looking forward to watching some other presentations.